So let's talk about exercise for the gram stain. So we basically have, um, through bacteria, I guess, or out of bacteria, two primary populations of bacteria. One is gram positive, one is gram negative. Now, physically, when you look at them, they are very different from each other. And the fact that they are different from each other means that we can stain them to distinguish between the two of them. So when you look at a gram positive versus a gram negative bacteria, we talked about the cell walls and they're made out of peptidoglycan. Now, if you look at gram positive, there is a very thick layer of peptidoglycan making up that cell wall. Sorry. So, this huge, thick cell wall physically is different than my gram negative. See this pink line right here? That's all the cell wall you get with a gram negative. And there is an extra membrane, phospholipid bilayer membrane, outside of that cell wall. This difference in structure actually allows you to basically take advantage of this different structure to stain them through a process called gram staining. Now, gram staining is a technique that we use to distinguish between gram positive and gram negative. Um, when you do this experiment, if you've got, new cells, you want them to be as fresh as possible. Now, why do you want them fresh? Because with some cell lines, you actually can switch back and forth between gram positive and gram negative, at least as far as their staining. So you want to make sure that you've got fresh cultures so that you don't get this false positive or false negative, okay? Now, when we do gram staining, as you saw in the previous video, we kind of have four things that we need. We need crystal violet, we need grams iodine, we need a decolorizer, which is basically alcohol, and we need safranin, which is a second dye. Crystal violet is one dye, safranin is another. So when we start, you're going to do a bacterial smear. Same as last week. It's same procedure. Everything is the same. Then you're going to take your sample and you are going to douse inside of that circle where you put your bacteria. You're going to douse it with crystal violet. Crystal violet sits on it for 1 minute. Now, crystal violet works its way into the peptidoglycan layers. Okay? Now, remember, if it works its way in, that's a lot of crystal violet that can stick inside of here. And there's going to be some crystal violet here. Now, after it sits for 1 minute, you rinse it off with water. Then you're going to put grams iodine. Grams iodine causes those pieces of crystal violet to stick together, almost to weave together. Now imagine I've got a ton of crystal violet in this peptidoglycan layer that's like that thick. And it's starting to bind to each other. That's going to be real good and anchored. Okay? It's going to stick to this huge peptidoglycan layer really, really well. Here, however, it's very thin. There will be some, but it's probably not going to be as much. And even if I get it to bind together, it might be bound to one crystal violet molecule here and one out here. So it's not necessarily as stable. Okay, so you put iodine for 1 minute and then you rinse. And then you put decolorizer onto the slide for 12 seconds. It's really important. It's 12 seconds. Why? Okay. The decolorizer is going to take this layer off of my gram negative, and it's going to take the crystal violet out of this peptidoglycan. Because like I said, it's really thin. It's not going to be anchored very well. Pretty easy to take out. Now, 12 seconds. If you leave it too long, it will also take the crystal violet out of here, regardless of how well it is embedded. Okay? If you leave it too short of a time, it will not have time to take it out of the gram negative either. So then you have two sets of crystal violet stained purple cells, which doesn't help us to distinguish between one and the other. So that 12 seconds is literally the right amount of time to get it out of here, but not out of here. Okay? So you got your crystal violet, you got your decolorizer, 12 seconds, rinse it, rinse it, rinse it with water. Okay? Now, the fourth element is the safranin. Safranin is a pink red dye. Okay? Crystal violet is violet, it's purple. And then you've got the safranin. So, safranin, you put it on top of your bacterial smear and you let it sit there for 30 seconds to 45 seconds to soak into our recently decolorized gram negative bacteria. So it gets into our cell wall. Okay. Now, this will also pick up the safranin. It's not like it's not going to, but it's already purple. So if I put purple and pink together, what color is it going to be? Purple. So even if the gram positive pick up the safranin, it's not going to matter because the darker color, the purple, is what's going to show up. So when you're done with that and you rinse it off and you dry it with your blotting paper and do those steps, you should have any gram positive purple and any gram negative that you see pink. Okay? So when it comes down to it, just like with the initial slide, when we talked about making the smear, I warned you guys about making it too thick, right? Same. Anytime that you make a bacterial smear, you have to be careful that you're not putting the cells on so thick that you won't be able to see through them. Now, this is especially important when it comes to the gram staining process. Even if you are looking at only one type of cell, let's say you're looking at a gram negative cell, you don't know it, but you're looking at a gram negative cell and you go through the process of gram staining. If you have cell layer on top of cell layer, there's a real good chance that crystal violet is going to stick in there. And if it sticks because you've got stacked cells, you're going to get a false result of having a gram positive when you actually have a gram negative. That's one of the reasons why I say you don't need very much. Just a little bit, it'll do. It's fine. So, when it comes down to it, any bacterial smear that you make, you got to be careful how much bacteria you're putting. So I hope this helps. And you've got the procedural video from Mr. Secord before this. Yeah,